Okay, so for the first chapter in this series, we're going to look at how you get a protein from a gene, bearing in mind the fact that this might be a little bit misleading in a title. We're actually looking more to get a polypeptide, and the polypeptide might be a gene, might be a protein, excuse me. So remember, it's DNA to RNA, RNA to the polypeptide, and the polypeptide might be a protein. So to do that, we need to look at two things. You can see them here, transcription, translation. Now, these are two independent processes that connect together to give you the polypeptide, which again might be the protein. If somebody ever says this expression to you, a gene expression, is saying how DNA shows up, how it does its jobs, and two jobs here that we know, of course, making itself, the S phase of interphase, and that's from mitosis, meiosis, and things like that. And then, of course, making the RNAs. And by making the RNAs, you're off to make your polypeptide. So this is specifically the making of the RNA. And then this is a great word for it, how DNA and the language of DNA is being made into the language of the polypeptide, which again, we hope is a protein. So all of this goes back to research. And even back in the early 20th century, this is still roughly 50 years before the discovery of DNA, they're already noting that genes, this heritable material, might be connected to your phenotype, and how you look and how you talk and how you walk and how you write and these kind of things. Please don't forget that the environment influences these things as well. But they are specifically looking at this heritable material and what it means towards the phenotype. And they thought that, hey, maybe if we alter this heritable stuff, it can cause a problem. In this case, something that's passed on that's a problem. And so they were looking at enzymes. And enzymes, of course, we know are a kind of protein. And they did see that it had connection. So they called it this one gene, one enzyme hypothesis, that if you alter the DNA, and notice how they were doing it, bombarding it with high energy radiation, creating a mutation, that it might affect the phenotype. In this case, the production of an enzyme, production of a certain kind of protein. Now, bear in mind, mutation is just change in the DNA. Sometimes it's good, sometimes it's bad. The vast majority of times it's silent. But in this case, it actually altered. And so they made that connection, yeah, that this heritable material therefore can alter the enzyme. But like I just said, bear in mind, not all proteins are enzymes. It's one kind of protein. So it had to be rewritten. And they did. They went to one gene, one protein. But again, like I was saying right at the beginning of this small lecture, polypeptides may not be a protein. So it again got rewritten. One gene, one polypeptide hypothesis. That if you alter the gene, you might alter the polypeptide. The polypeptide might be a protein, and the protein might be an enzyme. That's kind of how the connection went. But it was still intriguing research. But again, this is 1902. We haven't even discovered DNA yet, so it takes us time to get there. Now, flash forward, you get toward the 1970s, 1980s, 1990s. We're really understanding then how kind of DNA does what it does. And now we know there's these two parts, transcription, the making of the RNAs from DNA, and then translation again, going from the language of DNA to the language of your polypeptide. And as we've seen all semester, that happens at a ribosome. Just don't forget, a ribosome, they're two blocks of our RNA. So we're still dealing with RNA here. And therefore, we're going to focus again on how you do that. Yes, prokaryotes and eukaryotes have similarities, and there are also differences. Campbell doesn't really specify the differences, and that's okay, just as long as you have a major overview of how you go from DNA to the RNA to the polypeptide, you'll be fine. Now notice this idea of mature. So right off the bat, you're going to go from DNA to pre-messenger RNA, and that pre-messenger RNA has to be processed into mature messenger RNA. It's a mature messenger RNA that leaves the nucleus, goes down to the ribosome, it gets translated into your genetic transcript, your genetic output, the expression, which is going to be your polypeptide. So there you go. This is what you're first making then, your DNA to the RNAs. Your RNAs then move over to the ribosome, and the ribosome goes to a polypeptide, and your polypeptide might be a protein. This is, again, what's called that central dogma, and it's one of the two main jobs of DNA. Now, eukaryotes and prokaryotes, so this is prokaryote here, eukaryote here, have a functional difference. Don't forget, of course, that prokaryotes don't have a nucleus. They have a nucleoid region, yeah, where you find the DNA, but they don't have a nucleus. So it's like one to the next to the next. You go to your mRNA just goes right to the ribosome, and you're on your way. Remember, bacteria can have ribosomes. They can't have membranous organelles, but this is technically speaking a non-membranous organelle, so it's fine. And then your eukaryotes, of course, you have the nuclear envelope, and that's where a bit of an issue comes in. We've talked before how DNA is just enormous in size and can't get through the nuclear pores. So when we were talking about mitosis and meiosis, how the nuclear envelope has to break down, and now what is effectively inside is out. Well, in this case, then, you don't need to do that. You just need to go from your DNA to the pre-messenger RNA, process it to mature messenger RNA, and then that is small enough to leave the pore, we think chemically get attracted to the ribosome and get made into the polypeptide. And I like how they wrote polypeptide here, not specifically protein. Again, we're making a polypeptide, which might be a protein. So this has to be coded. Your DNA is the coding. One thing I do like out of like sort of traditional sayings is that DNA is a blueprint of the cell. Yeah, okay, that's fine, because it's coding for the RNA. And by coding for the RNA, you're coding for the sequence of amino acids. And yes, there are already 20 naturally occurring amino acids, which we've talked about before, all the way back in the beginning of the semester. Now, it's a language. Unlike English, which has single language words, so we have like an A as a word, an I as a word, even O is a word, like in poetry, you know, oh, what a beautiful day it is, that kind of idea. We have single letters that are words, not so much here. A single base by itself doesn't really mean anything, but groups of them do. So a group of 3 DNA bases could be called this triplet code. Then you can look at a group of 3 mRNA bases and a group of 3 tRNA bases. So this grouping of 3, that is the language by which you're going to code from the DNA to the sequence of amino acids for your polypeptide, going down here into the translational part. So the overall process then is your DNA. Now only one side is going to be used. So this would code for a different amino acid sequence than this side, coding for a different amino acid sequence. So notice, and don't forget, that in RNA, there is no thymine. It's replaced by uracil. A normally codes for T. So you would expect to see a T here, but notice we see a U. So we know the top side's what's coding. A for U, C for G, C for G, A for U, A for U, and so forth. If this bottom segment of DNA was being coded, well, then you would see an A here, then you'd see a C here, then you would see a C here, and so forth. So we know the top part is what's coding for this sequence of amino acids, or for this polypeptide. So our directionals are so important here, always growing in the 5' to 3' direction, as you saw, 5' to 3' direction, which then once this gets to the ribosome, will then be translated into your amino acid sequence, which will be your polypeptide, and again, that might be a protein. So this is a lovely figure to show you like the big picture. So there are 2 strands, excuse me, and then one called the template strand. This is the one that's used for ordering the sequence, the complementary strand of nucleotides. The other one is called the coding strand. So with this, we have our triplets, groups of 3 DNA bases that are giving rise to the mRNAs. And the mRNAs are also grouped in 3 bases, which are called codons. And then in the tRNA, they're called anticodons. So it's going from the triplet code to the codons in the mRNA to the anticodons in the tRNA. And that's going to give us our polypeptide sequence, linking together our amino acids. Ribosomes are amazing. They can actually group together any sequence of amino acids. It all depends on what you feed into it. It often reminds me of, think back like to the 1980s, 1990s. I know that's old tech, but a cassette deck. I still see them around sometimes. So you have a tape that goes through the magnetic heads, and the magnetic heads on the cassette deck reads it and makes it into music. It's like voltage, 0 and 5 volts, that kind of idea, and it makes it into sounds and music, that kind of idea. This is sort of the same thing, where this strand of mRNA, this one here, it's almost like a piece of tape being pulled through the ribosome. And as it's read, it's saying, okay, I need this amino acid, then this one, then this one, then this one, then this one. And it's linking together your amino acids. Again, the ribosomes can read any piece of tape. It just depends on what you feed into it. So there are a total of 64 codons then that can be sequenced for by the DNA. And therefore, it all depends on what segment of DNA is being read. And even in the segment of DNA being read, what's being viewed as a coding region and a non-coding region of DNA. We'll see that in the next chapter. So of these 64 codons that can be sequenced for, notice 61 of them actually make amino acids, but there are a few stop codons. These stop codons are just that. They're like a period at the end of a sentence. A period is not a word. It just says stop. That's all. It's kind of like that. These stop codons do not code for amino acid. They just say stop. In fact, a little like signal protein, signal polypeptide signal particle, there we go, that's the word I'm looking for, signal particle gets put in, the water molecule gets put in, and it stops. Particle, that's the word I was looking for. And so the sequence that's being read is called the reading frame. Now you can shift that reading frame, and if you shift it, you might get a different sequence that gets read for. That might happen if a mutation happens, which we'll see at the end of this chapter. So this is called a codon table. It all depends on what triplet is being read, therefore what codon is going down to the mRNA, and therefore going down to the ribosome, that's getting read out. So let's say that the ribosome is reading, I don't know, UCC, just off the top of my head. So what you do, UCC, the first base is U, so you go right here, this is the first one, so it's U. The second one is C, so you know they're coming together here, so I look for UCC, it's right there, which means the first amino acid is going to be serine. Let's say another one is, I don't know, AGU, okay, so the first one is A, the second one is G, the third one is U, AGU, serine again, what do you know? So it was random, I picked that. Let's do another one. How about GAA? So the first base is G, second one is A, so they come together here, GAA. And there you go. That's the amino acid that's going to be code for. It's very easy to use a codon table. On your test, you will not need to memorize this table. I will give you the table. What you do need to remember is two things: how to use it. First base, second base, third base. You're looking to see where they all come together. And then that AUG is a start codon. Now notice AUG must be at the beginning of the sequence, specifically for like 1 million sequences. And it always codes for methionine. Even though AUG must be at the beginning of the sequence, you can also get AUG anywhere in the sequence. It could be in the 5th spot, the 10th spot, the 121st spot. It just has to be in the first spot. And it will always give you methionine. And then you have 3 of these stop codons. Remember, they don't code for amino acid. It's that little particle plus a little water molecule. So the moment the ribosome reads one of these codons, UAA, for example, boop, that's it. Pop in that particle, you're done, you finish it out. So you need to remember that. You need to remember what the start codon is and the 3 stop codons, as well as how to use the table. Again, I will give you the table. As long as you know how to use it, you're good for the test. So with that, we've played, scientists see things they can do. For example, they got a tobacco plant to glow, so they actually grafted in the gene for fireflies, like sequencing for bioluminescence. Pretty cool. And then this one, it always gives me a chuckle to see this. Dr. Seuss, I'm sure, would be just tickled to death that we actually created a green pig. What do you know? So it just reminds me of green eggs and ham, that's all. You can see a normal pink pig behind it. One little side note here, a little bit of a personal annoyance. I don't know about the current edition with Campbell. This is like the edition before. We don't call jellyfish anymore. We call them jellies because they're not fish. They never have been a fish. They probably never will be a fish. So again, be careful with your science. It's good to be a little more specific than that. But still, they took the DNA from the jelly, stuck it into the pig, and they got a different expression out. So again, it all depends on what you're feeding into the system as to what you get out. Okay, so you have your sequence of DNA, and the DNA has to be told what to do. DNA just doesn't do it to do it. So it's going to work with things like RNA, and it's going to have this enzyme, RNA polymerase, is going to bind to it. And it's pretty much like the start here kind of molecule, and it's binding up towards the promoter. Now notice we're talking bacteria there. Starts in the promoter, and that's upstream of what you want to code. You code down the DNA, and you hit the terminator, and you stop. And once you have your ending product, this is your transcription unit. This is what's going to be coded out into your polypeptide. So there is the promoter region. It's like that green thing in there. There's actually something in there called a TATA box. There's the enzyme. That's pretty much the start here. Notice, like I said, it's upstream of the piece of DNA that you want to code. It's going to code down the DNA until you reach the termination. So it's promoter, the start, elongation, you open it up, and notice only one side's being used. You code, you code, you code, you code, and then you get to the termination. And once you're there, you have your transcript. This is the part that's going to go to a ribosome, be read, and made into your polypeptide. So you have these 3 parts: start, code, stop. It's really that direct. Now, as I noted to you, this little green piece, go all the way up to the top here, has a little, like, again, start here part. It's called the TATA box. TATA box. They note eukaryotes, but I've actually read some information that it can be found in prokaryotes as well. But that's okay. Campbell went with eukaryotes, so we'll stick with eukaryotes as well. So you actually have a whole complex. It's like a whole machinery that comes together. It's the actual protein itself, the enzyme that comes in, the transcription factors that come in, and then they bind all together at the TATA box, and then you start your coding sequence once you hit the actual like coding region of the DNA. So that's the complex that you're forming. You see it here, TATA, right there, TATA, TATA down there, TATA. Remember, this strand would code for a different polypeptide than that strand. Here's your transcription factors. There's the polymerase. Now you have the whole complex ready to go. The DNA is opening up, and you're using, of course, 1 or 2 sides of the DNA to code. It's going to code all the way down until you reach the termination. So you elongate, and you elongate quickly. Notice it's 10 to 20 bases upstream. Eukaryotes, you're coding 40 nucleotides at a time. That's actually pretty quick. I've read that bacteria is even faster. Of course, the faster you go, the more of a chance for a boo-boo, a mutation. But again, you code all the way down to you reach the termination sequence, then you stop, and you're still always going in the 5' to 3' direction, which is why it's being added to the 3' end. So it just gets read. The DNA is read. Notice this was a T, you plop in an A. This is an A, you plop in a U. Don't forget, there's no thymine in RNA. G for C, G for C, T for A, T for A. Again, A would have been T if it's DNA, but this is RNA, so you plop in a U. And you continue all the way down to end up in the termination. So again, it terminates a little bit differently in prokaryotes and eukaryotes. You sort of reach this terminator and you stop. Once you stop, it releases a little bit upstream. And then you have these, like, for eukaryotes, you might have these like little ends you stick on, the poly A tail, and then this like 5' cap. So poly A tail is a polyadenylate sequence. These help to like protect the mRNA as you go from the nucleus down to your ribosome. So what you created in eukaryotes, particularly again, is the pre-messenger RNA. So as I noted to you, you have to process this. There's 2 parts to the RNA, what are called introns and exons. You don't want the introns, that's the non-coding region, even though the vast majority of DNA is non-coding. So you get the introns out, you put together your exons, and you're on your way. This is known as splicing. And you can do this in a variable way, meaning in one case you might consider this an intron, in another case you might consider that an intron, which later on you'll see is called alternative RNA splicing. So as I mentioned, then you're processing this, and you put on that 5' cap and the poly A tail. So there's your 5' cap and the polyadenylate tail. This helps to protect the DNA. It's going to be the RNA, I apologize, because you have to go from the nucleus into the cytosol and from the cytosol down to the ribosome, and that's kind of a rough environment for this RNA to get through. So it helps it not to degrade. UTR is the untranslatable region. The UTR kind of dictates how long this is going to hang around until this begins to degrade out. You don't want to be coding for it forever and ever and over. You'd be wasting your resources. So like I said, you have these introns and exons. There's a really easy way to remember this. Think that exons exit the nucleus. So you want to get the introns out of the pre-messenger RNA, glue together all the exons, and then send the mature out. That is indeed the RNA splicing. And as I noted a moment ago, it can be a little variable what the cell is considering an intron and exon, which means you can stick a word in here called alternative RNA splicing, and you'll see that momentarily. So here is your pre-messenger RNA. It's got the introns in it. You don't want those. Those are non-coding regions. So you pull them out, and then you glue everything back together again. You glue the exons back together again. Again, exons exit the nucleus. That would get sent down to the ribosome, and you process into your polypeptide. Now, there are 2 ways to get this stuff out. Spliceosomes are these small nuclear ribonuclear proteins. They're little proteins that cut and then kind of glue together what you have. So here you go. Here's your exons and introns. It kind of creates this little bow. You put together the introns, it cuts it out, and then you glue together the exons. Kind of cool. Then you have these things called ribozymes. This is, I know I'm a raging nerd, but this is incredibly exciting because I mentioned to you way back when that I said enzymes. Enzymes used to be called biological catalysts. And I said something like, please don't call them that. Call them a kind of biological catalyst. Because today we know other things can function as catalysts, not just enzymes. And in this case, RNA is working as a catalyst. And that's really consequential. Not so much maybe in Bio 1, but certainly in Bio 2, because we have to deal with the idea of, well, certainly evolutionary theory, but how do you go from nothing to something? So how do you go from there was no planet, then there is a planet? Of course, that's the accretion hypothesis, swirling dust and gas, got the heavier stuff at the interior of the planet, lighter stuff at the exterior, but it was a molten ball of goo, the planet. Then it cools down and has liquid water on its surface at about 3.8 billion years ago, but there's still no life. But then certainly by 3.5 billion years ago, there's life showing up in the fossil record. So I've seen the data a little bit different. Is it 3.7 billion years ago, 3.5 billion years ago, the life is showing up in the fossil record? That's debatable, but still by 3.5 billion years ago, there's life in the fossil record. So how do you get there? How do you go from nothing to life without violating spontaneous generation? And that's particularly true that all life on this planet uses genetic material, whether it's RNA or DNA. Well, that begs the question right here. How do you go from nothing to something super complex like DNA? You can't just jump there. That doesn't make sense. So we think we actually got there from RNA. And in fact, there's an argument on this planet that this planet is an RNA world, not a DNA world. What do you know? So it's interesting because then we know now, one, RNA is small, it's 70 to 90 bases long, it's single-stranded in nature, and it can be catalytic. Not only that, you can get RNA to produce things. So again, could we go from RNA to DNA? We can, and we know we can because of the retroviruses. Things like HIV does go from RNA to DNA. So this becomes really consequential that RNA can work as a catalytic molecule. And that's what you see here, that it's being able to use to kind of process out this material. It is a kind of biological catalyst. Very, very exciting that we have that. Okay, there we go. With all that, now, like I said, what you use as, and therefore what your cells seen as an intron versus exon can be a little bit variable. So in that case, it used to be thought, like with humans, oh, look at us, we're so complex. We must have 100,000 genes. We must have a lot going on. When we studied it, we found out that humans only have about 20,000 genes, roughly about the same number of genes as a mouse. Not meant to be in criticism towards humans or a mouse in any way, shape, or form. Mice are complex. But again, it was with this idea of, well, wait a minute. How are you getting all that complexity with only 20,000 genes? Well, it's a lot of variability. You can do a lot with a little. And so it all depends on what you're considering to be an intron versus an exon. And there's, again, a little bit of like malleability there. And then, of course, you can group. You can have structures that come together to give you like an overall bigger idea. So this exon shuffling resulted in this. And evolution, again, just means change over time. And when you get this new and novel product to be formed, then natural selection can work on it. I know I mentioned that before. I don't know who your Bio 2 teacher will be, so bear that in mind. Evolution does mean change over time, but evolution tinkers. It doesn't necessarily create. You already have to have it in the population. Again, Charles Darwin was very clear on that. This happens at the populational level. Evolution doesn't happen on the individual level, although Dawkins in the later 20th century did question that idea. But either way, that evolution has to work on something already there. So once you get something new, like these new proteins, now let's see what that does. Does it confer an advantage or not? So this idea of shuffling is really quite intriguing. What are you considering exons and introns? And then how are you grouping things together, this modular design? Now, with that all in mind, now we can actually talk about making your polypeptide. So you go from DNA to the mRNA, mRNA to the ribosome is reading the mRNA, but you still have to have these guys come in. It's a perfectly named molecule, transfer RNA. What is it transferring? A single amino acid. Now, DNA also codes for tRNA. Don't forget that our DNA codes for our RNA, and our RNA, as we've spoken about before, is of course your ribosome. So this is still being coded out by the DNA, but this is like in the cytosol now, and it could pick up an amino acid. Where do you get your amino acid? Your diet. We've talked about that before. Never underestimate the power of your diet. So it picks up the amino acid, and then when the ribosome reads the codon and says, oh, I need this tRNA, please, thank you very much, that tRNA will bring its amino acid in and put it into the polypeptide sequence. So this is an entire ribosome. The ribosome has two parts to it, two blocks of rRNA. It has the very fancy name of small ribosomal block and large ribosomal block. And then these three little gaps, I'll come back to those gaps in a minute. Now notice, these are uncharged tRNAs. They haven't picked up an amino acid yet in the cytosol. Once you've picked up an amino acid, like whatever this small blue circle is, that's going to be a charged tRNA. And then when the codon is read by the ribosome, it's saying chemically, I need this tRNA, and this tRNA comes in. So it comes in, then you have this little peptide bond that's going to form between any amino acid that was there beforehand. You can see my cat is, it's a chilly day. My cat is stealing my heat, but he is very sweet. And so please forgive his arm. But anyways, there is a peptide bond that forms, and then translocation happens. All of these amino acids are given over to this guy. Everybody bumps on the line. And as you can see, as it happens, the uncharged tRNA is kicked out back into the cytosol where it can maybe pick up another amino acid. So this is what tRNAs are used for. Now, tRNA has a long sequence to it. It's just a long sequence of bases, but you notice right here, and it's a little tiny, looks like CCG, that is called the anticodon. So we have the triplet code of the DNA, the codon of the mRNA, and the anticodon of the tRNA. So it really is, this codon is saying, I need this anticodon, which is what's chemically calling for this specific tRNA to bring in its specific amino acid. There you go, the anticodon. So tRNA is pretty cool. You can see it's this long sequence of nucleotides. This is the top part that would hold the amino acid. That's neat, but this is the part we're really focusing on. This is the anticodon that's chemically being brought in because of whatever codon is being read by the ribosome. Now you can see there's different shapes to it. This is called the cloverleaf shape. Maybe it looks like a cloverleaf to you. This is called the bent L shape. It doesn't matter. It's just the shape of the DNA. And notice that in the bent L shape, they're focusing on, of course, the anticodon, which is what we need. So there is your bent L shape. Now, there's like a bonding that goes on here. So the matching here is done by this enzyme, amino acyl tRNA synthase, that's helping to connect the tRNA to the amino acid. There is a little bit of flexibility. I like how your author notes that, a little bit of wobble here. Okay, so let's go back real quick from this slide. Okay, so slide 35. Let's go back to that codon table for a second. Okay, so when mutations happen, mutations are at the end of this chapter. You have to ask the question, is it a front-loading or back-loading mutation? So notice that leucine, there are 4 different codons that give you leucine. So that wobble idea, if it's a back-loading mutation, let's say it was supposed to be in the DNA sequence coding for guanine, but something happens, you get like a point mutation, and now it's coding for cytosine. Notice so you still get leucine. That's wobble. You had a little bit of mutation, but it didn't affect the polypeptide output because it didn't affect what amino acid it was coding for. That would be a silent mutation. That's a back-loading mutation. But let's say it's a front-loading mutation, and instead of getting CCU, now you get GUU. Okay, so instead of getting leucine, let's see what GUU is. GU, so it's here, GUU. Now you get valine. So you still get an amino acid that's in the sequence, but now you might get an entirely different polypeptide, entirely different function. Maybe it's beneficial, maybe it does nothing, maybe it's a deleterious mutation. You have to see what happens. And then, if I may put it this way, heaven forbid, you get one of these mutations that goes from an actual coding sequence to one of the stop codons, you might kill the sequence. And who knows, maybe even like amino acid number 2 had a mutation. You would have had 150 amino acids, but in slot 2, the mutation now codes for a stop codon. You don't have a polypeptide, you have 1 amino acid in the sequence. So this is that wobble idea. Go back to slide 35. This is that wobble idea, where you can get a little bit of flexibility in what's being coded for, but it all depends on where that mutation happens as to what codon you're getting, therefore what amino acid you might get. And if you code for a stop codon, look out. Okay, so you have the uncharged tRNA. You need to bond it together. What do you know? ATP. How many times does ATP come up in this course? Absolutely amazing. And ATP is going to bind with this, this protein type. So you have the enzyme, the synthetase, comes in, now you have the tRNA, you have the amino acid, it bonds together with this enzyme, the synthetase, and you get the charged tRNA. This is the charged tRNA that would go down to the ribosome and bond to, if you want to put it that way, bond to the codon. I put bond in quote. Remember, the only peptide bond there is forming is between the amino acids, but you're dropping off the amino acid. Okay, so like I said earlier, our ribosomes come in 2 blocks, the small ribosomal subunit and the large ribosomal subunit. Once you have that, you have your ribosome, and then it's reading the codon, codon. And as it does so, it's bringing in the tRNAs, and it'll do so until it reaches a stop codon. Yes, this is also a nice little note here by the author, just to remind us that ribosomes do vary between eukaryotes and prokaryotes, and it's one way we could target medicines, these antibiotic drugs, so that it would hurt, say, a bacterial's ribosomes, and therefore probably kill the bacterium, but doesn't bother a eukaryote ribosome, therefore thankfully not killing us. Kind of nice. All right, so you have those 3 sites I talked about. This is the peptidyl site, amino acyl site, and the exit site. I know those are crazy names, but let's see, there it is. So your A site, amino acyl, the P site, peptidyl, and then E for exit. That's kind of nice. Notice that groove right above the peptidyl site. So the mRNA is going through, it's being read codon by codon. The new tRNA, the one that's charged, comes into the amino acyl site, comes into that amino acyl. and it's bringing in its amino acid. When it brings in its amino acid, all amino acid before it get bonded over to peptide bond to the incoming amino acid, and then it translocates over to the incoming tRNA, everything bumps down the line. This now has lost all of its amino acids. They're all bound to this guy. So it goes into the E site and gets kicked out. This guy is now holding all of the amino acids and goes into the P site, and that opens up the A site. The next codon is read, and the next sequence comes in. So initiation is the first amino acid coming in, the first codon being read. Remember, methionine, the start codon. Elongation is actually what I just described, how everything is bonding and bumping down. And then, of course, you terminate when you hit a stop codon. So again, the mRNA gets chemically attracted to the ribosome. It actually binds to the small unit and starts getting read, and then the large unit slams down on top of it. So it pretty much goes right to the P site, freeing up the A site. And it's being read at the start codon, that's AUG, bringing in, of course, methionine. And then once that tRNA has come in, then it reads the next codon, which is now in the A site, bringing in the next tRNA, its amino acid. So there you go. There's your small ribosomal subunit. There's the tape, your mRNA, and there's methionine. Once that's been read and that tRNA is in, then the large block slams down on top of it. And you can see that here, like I just said a second ago, it's already in the P site. So the methionine is there, means the A site is free. That codon can be read, and the next tRNA can come in. That's going to be your elongation. And Campbell did a good job here, noting that, of course, elongating and growing the chain on the C terminus and on the chain. Remember, these are amino acids coming in. So amino acid 1 linking to amino acid 2, now you have a dipeptide. Amino acid 3, a tripeptide, amino acid 4 and 5 and 6 and so forth, until you reach your stop codon. Always growing in your 5' to 3' direction, creating those peptide bonds as you go. So right here, there's your P site. You can see it has its tRNA there. It looks like 1, 2, 3, 4 amino acids have already come in. So the A site's open. It's reading that codon. Here comes in the next tRNA. So the anticodon being coded by the codon comes in, and it brings in its amino acid. Once it's in the A site, all that was here before, now you create a peptide bond, and they all translocate over to this tRNA. Once that's happened, you can see that there. This guy bumps into the E site. This whole thing goes into the P site. And once that happens, the uncharged tRNA is kicked back out on the cytosol. The tRNA that's now holding all of the amino acids, should be 5 now, 1, 2, 3, 4, 5, is in the P site. The A site is now open again, ready for the next tRNA to come in. You're going to keep doing that again and again and again and again and again, and that's really quickly, mind you, until you reach a stop codon. And then again, that little particle goes in, it's the release factor. Some sites like to say a little water molecule comes in, everything kind of breaks apart, and now you have your primary sequence, something we talked about way back at the beginning of the semester. You still have to go through the secondary and tertiary structure at least, and things for like hemoglobin would be a quaternary structure, but at least you made your primary sequence here. So there you go. In this case, it's reached the end. There's your release factor, the little particle I was talking about. Everything kind of dissociates, and you have the primary sequence. Now it needs to be folded up, mind you. So this either has to go into the endoplasmic reticulum or to one of the chaperonins that we talked about way in the beginning of the semester to go into your secondary and tertiary sequence at least. Now, we have found out that you can make a lot of protein really fast. So you can have many ribosomes pretty much all working on the same mRNA, and it is indeed called a polyribosome. So what a great micrograph, huh? And you can see, you can actually make out the mRNA, those strands in between all of these blobs. These blobs are your ribosomes. So they're just churning out proteins. They're just churning out proteins as that mRNA is being read through. But at some point, it has to stop. You don't want to make too much. You don't want to waste your resources. So that UTR region, the untranslatable region, will help you out with that. So like I said, once you've made your primary sequence, then you got to process it. You've only made the primary sequence. So like I said back in the day, you got to take stuff off, put stuff on, fold it up, that kind of idea. One of the intriguing things with mammals is that even though the start codon, the very first amino acid in the sequence is methionine, it is not unusual for, in the post-translational modification, to get methionine, the first amino acid, to be ripped off. So in a future class, for example, if you're reading an amino acid sequence, from like amino acid 1 to, I don't know, 120, and you're saying, wait a minute, this is a mammalian sequence, and it doesn't start with methionine. Oh, yeah, that's right, because it went through post-translational modification. Kind of keep that in mind. You got to process this, but you have to process it somewhere. Remember, you either have to go into the endoplasmic reticulum, so that'd be a rough endoplasmic reticulum, and therefore your ribosome would have been bound to the RAR, or it would have been at a chaperonin. So that's what this is. This is noting that you have two places to find ribosomes. Either, again, free ribosomes, I love it, free ribosomes, or like free-ranging ribosomes in the cytosol are bound to the ER. If they're bound to the ER, you've actually got to do a little bit of work to get the ribosome to attach to the ER, and then for the growing polypeptide to grow into the cisternae, the channels inside of the RER. And it looks a little bit like this. You need this, a single peptide. So this is like a little end of the like amino acid sequence you made, and it binds to this particle. This particle then helps it bind to the ER, and then as you continue to grow the polypeptide chain, it grows into the ER. So there's that signal particle, a little peptide. There's the particle, excuse me. Notice how this little peptide is part of the growing amino acid chain. The particle attaches to it. Now that it's attached to it, then you can get the ribosome to be bound to the ER. And the growing polypeptide chain grows into the cisternae. Now, once it's in the cisternae, this lumen, you can process it in the ER. Don't forget, once it's done in the ER, it gets into a vesicle, gets sent over to the Golgi for final processing. Last but not least, in this chapter, mutations. As I mentioned, mutations, don't define them as good or bad. It's just a change of the DNA. The vast majority of mutations are silent. No effective change of the polypeptide output. Some mutations are not good. Some mutations are actually beneficial. I noted too that evolution actually works on mutations and things like gene drift. These are some of the driving forces of things like natural selection there for evolution. So mutations, again, you have to see what they do. But because some can be bad, you don't want to go out and like look for mutations. You don't want to go sit in the sun and say, I'm going to mutate. You don't do that to yourself. You'll just end up with skin cancer. So there's a number of different kinds of mutations. Point mutations are just that. It's a single base change leading to maybe a single amino acid change. We already talked about one specific kind of this. That was sickle cell anemia. That single base change gave you entirely different amino acid sequence because it was a single amino acid change, giving you those odd-shaped red blood cells. So again, we can see that a single base can really be consequential. So don't forget from a previous chapter, wild type means like what you're expecting to get. Mutant would be what you don't want to get or don't expect to get. It doesn't have to be bad. It's just what you're not expecting to get. Remember, sickle cell anemia and the heterozygous condition actually confers some advantage. Homozygous dominant doesn't have sickle cell, and homozygous recessive, that's a bad position to be in. But like I said, when you don't have the point mutation, you get glutamic acid. And when you do have that single base substitution, you get valine. Single amino acid change because of a single base change. And this creates those odd-shaped red blood cells. So it's pretty amazing. So there are different kinds of mutations you can get. Point mutations are one of them. They're just at a single base change. Now with that, silent mutations, again, no effective change to the polypeptide output. Missense, the reading frame has been shifted. It's like changing how you read a sentence. It might make sense, it may not make sense, that kind of idea. And then a nonsense, notice you plopped in a stop codon. And that could be absolutely devastating to your polypeptide sequence, especially if it's earlier in the sequence. Okay, so this is really tiny. The bottom slides are exactly the same. So let me show you the bigger picture. So insertions and deletions are just that. You insert something, you insert a base, maybe even insert an entire triplet. So you insert an entire codon, shifting what you're reading, frame shift mutation. And it looks a bit like this. So this is the wild type, what you should have gotten. Notice here you inserted a single base, which means you inserted a single base here in the mRNA. Now, in this case, notice you were coding for lysine, but now you popped in that stop codon. So no matter how many amino acids you would have gotten in the sequence, let's say 150 of them again, now you've got one. That's obviously not a polypeptide. You've obviously just ruined whatever this would have done. This could have been very consequential. Look out, absolutely devastating. So you have to see what it does. Okay, so in this case, a deletion. So this is the wild type, what you should have had. Now you had an A pulled out, so you pulled out. You would have gotten methionine, lysine, phenylalanine, glycine. Now you still get a sequence of amino acids, but they're different. Methionine, lysine, leucine, and alanine. Maybe this has still functionality. Maybe it even gives you better functionality. So this would be something that once it's in the population, the evolutionary sequence might favor it. You'd have to see. But then again, this could also be deleterious. You just have to see what the mutation outputs as. In this case, what you should be getting, and in this case, you pulled out an entire triplet, which means you pulled out an entire codon. And notice now you might have actually ruined the sequencing. It depends on when that stop codon comes up. So it all depends on what it does as to what the output is going to be. So why do these mutations happen? Mutagens, there's a whole slew of things that might be mutagens. Notice physical mutations, chemical mutations. So physical, maybe you're out in the sun and you get a thymine dimer from solar radiations, you get skin cancer. Chemical mutations, so that could be from smoking, from drinking, from food additives. There's a number of things that could cause this. The more processed the food, the more lookout it is. All those chemicals like red dyes and blue dyes and yellow dyes, these kind of things can be problematic. So this is why we talk about eating more natural foods. The idea of organic versus non-organic is a consequential thing here. The only thing I'll note on that, because it's not part of the chapter, is that organic should be more nutritious. It's not so much that it tastes better and hopefully leads to better health. But again, there's still a lot of research going on there. So it all depends on what mutates the DNA. We worry about this. Heck, I just saw something this morning. It was an article that came out, a newspaper article, talking about it was like a chemical additive for, it's like hairspray or something like that. And now a slew of people getting things like certain cancers. So we have to be very careful with this, things we put on our bodies, things we ingest, even the skin, don't forget, it's a living organ. It can absorb through the skin. We got to be careful about these things and what they might do to us. So the last but not least, again, goes back to evolution. And this is really segueing you over towards bio 2. So bacteria are in two forms, like the everyday bacteria that you just find everywhere, it's like covering everything. And then the extremophilic, stuff you find in rough places like methanogens, methane pools, and acidophiles, acid pools, and thermophiles, and hot water baths, that kind of idea. There's some ways that archaea are more related to the eukarya than they would to the bacteria. So this whole idea of like a tree of life, it's not quite right, where you start here and everything branches out. We look at it more like bushes of life and a lot of mix matching going on, especially with horizontal gene transfer. So we could talk about that here as well, comparing these guys. And it's not that easy anymore. There's some ways that the bacteria are different from the eukarya, but the archaea might be more like the eukarya. So right here, the archaea might be more like the eukarya than they are the bacteria. So it's not as easy just saying bacteria do this and eukarya do that. And I've been saying that since the beginning of the semester. It's not as cut and dry as people like to make it. So I also been saying since the beginning of the term, learn things in context, that kind of idea. And so that's really beginning to get to us to the point of asking, what exactly is this? What exactly is DNA? How exactly does it work? Now, up to this point, we've done this. We've talked about it being inheritance. You pass it from one generation to the next. We've talked about it being a specific sequence of nucleotides. Yes, it's a polynucleotide structure. Yes, and that it codes for DNA. But again, this isn't as direct as people maybe think. It's, as I think how I put it, it's a powerful molecule. It's not magical, but it's powerful. So here's the big shebang, DNA to the mRNA to the ribosome to get your polypeptide sequence. But I thought I would show a little video here for you to give you an idea. Let me transfer over my sound. Okay, so I can't talk during this, but hopefully you can hear it well.
The innermost working of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of biology. How DNA makes protein. It starts with a bundle of factors assembling at the start of the gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific three-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. Code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic method you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
And there you go. Pretty amazing. 100 trillion molecules a second. So this is really, really, really fast. Something is autoplay. We don't need an ad. There you go. So it's really, really, really fast. So I hope you enjoyed the chapter. If you have any questions, of course, feel free to let me know.